to start by thanking the organizers for bringing me to this very exciting meeting. It's really inspiring to see all the enthusiasm and human potential. It's my first time in India, but already a second meeting. So I'm coming here from an FDMM meeting in Kolkata, where we also heard a lot of sort of Indian science presented, and it was very exciting, and I hope it will continue. So I was quite humbled by this task of giving some sort of a mentoring talk, because I really don't think I'm, accomplished enough or qualified to really sort of mentor anyone. So what I thought I'd do first, I'd say a few words about my own mentors, because they were qualified, and I hope maybe I can sort of transfer some of this to you, and then to discuss briefly my own attempts to set up and run a lab, largely by negative example. And so I became interested in immunology as a medical student undergrad in growing up in the former Soviet Union, and I was extremely lucky to start my training in science with this person, Gary Abilev, who probably most of you are not familiar with, but he is a remarkably sort of accomplished scientist. He started his career in science as a graduate student by purifying histones. It was not because there was an iron curtain. The Western science did not learn about it in time, but he was really the first to purify histone as a protein. And then in the 60s, he made a real fundamental and widely acknowledged discovery of the first oncofetal antigen, alpha-fetoprotein. He showed that it was re-expressed in liver tumors, and actually brought this as a test into clinical practice. So this was widely acknowledged, and he was very much appreciated in the West. But his career was totally derailed by the Soviets, because he was Jewish and not a party member. So he was very actively discriminated against. He was kicked out of the institution that he almost founded, and he was prevented from going abroad and or meeting his colleagues in the West. So his career challenges were probably much more severe than anything we can think about. And then finally the Soviet Union collapsed, and then he finally started getting all the recognition that he deserved, but then his entire lab basically left because everybody wanted to go to the West. So he basically got his recognition, but pretty much his career was finished because of that. Despite that, he not only did not try to prevent his people from going abroad and getting the training and jobs, but he actively encouraged them. So including lowly undergrads like myself. So this kind of approach and support was really sort of impressive, and the selflessness there was very remarkable. And then I was lucky to start my graduate school at the Weizmann Institute of Science in Israel. There is a funny side story, because I didn't have any formal education at the point, but luckily Israel is a very informal country. They said, oh, whatever, just get started. And so I did my PhD with this man, Irun Cohen, who's a very accomplished cellular immunologist at the Weizmann Institute. And I'm actually showing this picture of him here with his family, because this was basically, it symbolizes him. Irun was all about sort of treating everybody like, and especially his lab members, like their family, and providing support and empathy like his students were his children. This was absolutely remarkable. He was giving students a lot of freedom, yet was there to support them, and was very generous. Just as a small example, I remember that I was running molecular modeling, and we had a facility for that, so we paid some significant money for them to model the protein I was working with, and then at one point I realized that I gave them the wrong sequence to model. So I was so kind of desperate that I called Yaron at home sort of to tell him about this disaster. He listened to me, and then he started laughing. Because for him, you know, this was not, you know, an issue. Support to his students was more important. And if I was ever able to carry some of this attitude in my own career, I think I'd be a happy man. And so Yaron was studied autoimmunity, and he was a very deep thinker, and I became interested in these questions of self, non-self discrimination and prevention of attack of the immune system on itself ever since. This is his favorite example. This is a sculpture called Kadishman Tree that stands at the Weizmann Institute. So he was thinking deeply about the representation of self in the immune system and how the attack on self is prevented. So I became very interested in this question, but I felt that my molecular training was not sufficient to address it properly. So I went to do a postdoc with Phil Leader, who was at the time was the chair of genetics at Harvard. And Phil started his career as a postdoctoral fellow with Marshall Nirenberg by deciphering the first letter of the genetic code and then 50-something letters of the genetic code. So that was his perspective on scientific research. Here he's shown receiving the Lasker Award for discovering VDJ recombination together with Tonigawa and Lee Hood. And the fact that not all of them proceeded to get the Nobel Prize for it is one of the grossest injustices out there. But anyway, this was basically, as you can imagine, Phil was very hard to really impress, right? Because that was his perspective on science. And yet, despite all his accomplishments and his high sort of stature, he was incredibly approachable and personable and generous person. Everybody called him by his first name. And he, again, was sort of deeply concerned about his trainees and was going out of his way to support them. Again, just one small example. At the time, postdocs basically had zero social protections in the US, and we didn't have dental insurance, among other things. And so he accidentally learned that I have dental problems, but I don't have insurance, so I just have to kind of suffer a bit. So he went into it. Then he went through all the options and realized that there is indeed no legal way to give me dental insurance. Then he just gave his fund account to Harvard Medical School and told me to go and get treatment there, and they would charge his research money directly. So this was the kind of mentor he was. And again, these are the lessons I've been learning from my mentors, that you can be as distinguished a scientist as he was, but also just a very noble and generous person and a good mentor. And so Phil, because of his career, was all about genetics. That was his favorite saying, and anyone graduating from my lab should swear on these words. So here he is shown with the so-called oncomouse, the first transgenic mouse that was developing tumors, which basically brought in the whole era of mouse modeling. And so that's the attitude that I sort of inherited from his lab and tried to carry forward. So in his lab, I was mostly studying the development of T lymphocytes. And well, even though it was kind of fun, by the end of your 5th year, you start feeling that maybe you had enough of T cells. So when I was lucky to get my own faculty position at Columbia University, I made kind of the most stupid decision to change gears. Not, at the time of starting your own lab, also to start looking into a different set of questions for which I had very little experience or qualifications. So this was the time when there was a lot of attention to dendritic cells in the immune system. So these are key sentinels of the immune system, conventional dendritic cells that present antigen and initiate adaptive immune responses, and plasmacytoid dendritic cells, or PDCs, that are sentinels that produce cytokines such as type 1 interferon. So I decided to change directions at the worst possible time, and at the same time as starting my lab, I decided to switch the focus of research. I wouldn't recommend it to anyone, honestly, but again, that's what it was. Then I started my own lab, and I think I made every possible mistake. Just to list some of them, I actually did not negotiate well because I was just so happy that somebody is giving me the opportunity to do research, right? It's totally naive and silly, but that's what it was. And then, inevitably mistakes started piling up. I showed up at Columbia University and realized that I have no tissue culture and no mouse space, right? Because I thought it was self-evident that I would need those, and they, of course, didn't bother. So that took some kind of pushing and negotiations, and luckily, eventually, things started sort of coming together, but it was a very rough start. The next thing I realized as we started running experiments is that nothing is working. Even things that I thought I can run blindfolded don't work, right? That was probably a consequence of the fact that I had a major sticker shock, right? Because then I realized how much things really cost, and I started saving. Well, you start saving, but then that comes at the expense of your experiments. So as we were discussing yesterday at the breakout session, sometimes it actually helps to buy more expensive things. Just think about it as an investment, because having your experiments working are more important. Well, eventually things came back together, but then the biggest challenge is to actually find people to work, right? And again, against the advice of my senior colleagues, I was so impatient to get the lab running, so that I quickly hired a few people, and not all of them were the right people, but as I realized later on, they are easy to bring, but very hard to let go for actually legal purposes, but also, of course, for emotional purposes and social. It really rips the social fabric. So yeah, nobody should be rushing to bring people. You need to bring the right people, which, as I learned through my own sad experience. And then I realized that I know absolutely nothing about how the administration and financing works. In Phil's lab, we really thought money just grows on trees. So I still remember standing in the administrator's office. She was explaining to me what a fringe benefit is, which is essentially a tax imposed by the university. So in addition to the salary you pay to your postdoc or student, you're paying extra third to the university, right? Somehow these little secrets are sort of hidden somewhere, right? So I had a very rough awakening to a lot of these kind of things. And then, of course, finally you start writing grants. And then they start coming back at you with some really kind of sharp comments. This is an actual quote from a review that I got, right? So things were getting rough after about 3 years of me being a PI. There were no papers, and I still couldn't get a major grant, so things were a bit rough. And so maybe the only thing that sort of really gets you through this and makes your life worth living is the young people who come to your lab. So in that I was very fortunate. This is my first crop of graduate students mainly, and a couple of postdocs. And that's really the best part of starting a lab, because young, enthusiastic people come to you. They know very little. For a long while, I was manually checking every primer sequence that they were ordering, because they were sort of making mistakes all the time. But the energy that they bring and the enthusiasm is really priceless, and eventually makes your science work. So things slowly started working out. Among other challenges we gave to ourselves was to make a mouse that would allow gene deletion specifically in dendritic cells in vivo, the so-called Cre-deleter strain, which was missing in the field at the time. And again, in retrospect, it was a very sort of silly decision, because every major lab at the time was trying to make one. And somehow I was arrogant enough to think that, I'm just going to do it. And somehow, miraculously, with the help of these enthusiastic people, it helped. As an immunologist, I have to show at least one flow cytometry plot. I'm sorry. You just have to deal with it. But it's not too complicated. You can hopefully see that you have major Cre activity in dendritic cells and very little anywhere else. So things worked out. And then, because I knew that it's just such a badly needed reagent in the community, I started sending it to other labs and collaborating with other people long before we actually published it, which was also a very risky decision. I almost got scooped by my own collaborators. But again, things worked out, and then, of course, I quickly deposited it in repository. So this became a standard in the field. There are about 300 papers on the Jackson Lab repository website that have used it so far. And so even in retrospect, I think it was a great experience and probably right decision to just share, because it's so good for the community and pushes science forward. So recommended. And then other things slowly started coming together. We really were able to work out the developmental sequence of these plasmacytoid dendritic cells, identify some of the key transcription factors, such as this guy here, E22, and in general kind of give a more thorough genetic and molecular description of dendritic cell development, hopefully using some of the training I got in Field Leaders Lab. So that's cool, and that's nice, but I have kind of a bit of a bad news for junior investigators here, because after this initial, several years, finally, funding comes, papers come. For a brief moment, things are working well, but then you quickly enter something that in the US is called the Death Valley, which means that now you have to sustain your lab. And that's a separate and very difficult challenge, right? Well, you get tenure, but this is a quote from my good colleague Avinash Pandula, who is now at the NIH, which I think is a very sort of accurate description of the process. You get tenure. In the US, it really means nothing. In other places like here, it may actually mean something, but you still need to run your lab. So not much is changing in reality. So you were not killed. That's pretty much the only thing. And then you are sort of, you don't get any discounts for being junior anymore. You're not eligible for all the junior awards. Startup is sort of ending up, and your major grants are up for renewal, competing against, the top guns. So things get difficult. you can no longer devote your full time to experiments and eventually have to sort of to stop, and then you become fully reliant on people in the lab, right? So your experimental work becomes as good as your people are, which is a very painful transition. Conversely, you have to deal with just with a lot of things that we'd rather not deal with, right? And that's progressively piling up. And you cannot run on the same ideas for the rest of your life, right? So you still need to come up with new ideas and directions, but you're not starting from scratch anymore. Your car is running, and you have to kind of change directions, maintaining it at the same time. So this is a new set of challenges that I'm just a fair warning to the folks there. And again, if anything makes it happen, it's your people. So this is my lab around that time. So again, I was fortunate to have very sort of enthusiastic people, including these two talented individuals, Dipayaman Ganguly, who is co-organizer of this esteemed conference, and Hiya Ghosh, who is now a PI at NCBS in Bangalore. So through the sort of combined efforts of this talented crowd, we were able to sort of to convert some of this early work that we've been starting, and in particular bring it back to the questions of function and autoimmunity. So again, using these very basic facts that we learned about the development of plasmacytoid dendritic cells, we were able to target them and learn more about their function, like in this study where we showed that in the absence of PDCs, mice are highly susceptible to certain acute viral infections, as well as persistent infections, such as chronic LCMV, which is a reasonable approximation of things like HIV and Hep C infection. There was a lot of discussion about basic versus translational research. We found, somewhat unexpectedly, that all these very basic questions about transcription factor machinery that is driving dendritic cell development got a very sort of unexpected translational application, because it turns out that, well, plasmacytoid dendritic cells have kind of an evil counterpart, a PDC leukemia known as BPDC, and highly aggressive, pretty much deadly tumor. And it turned out that these cells were essentially addicted to the same transcriptional machinery that sustained normal PDC development. And it was targetable. And this was probably the first kind of more or less rationally designed therapy for this otherwise deadly tumor. So these very, very basic questions found very unexpected but very important translational application. So I think as long as the science is good, the translation will come. I should also mention that this was a very productive collaboration with Lou Stout, a very famous lymphoma researcher at the NIH. So again, adding to the value of collaborations. And then finally, we were able to go back to the interest of my graduate days and get back to autoimmunity, in this case looking at the role of plasmacytoid dendritic cells in autoimmunity, and in this work by Dipayaman and Vanya, another postdoc in my lab. They were really able to provide solid genetic evidence that PDCs are required for the development of systemic lupus erythematosus in mice in 2 different models. Autoantibodies are wiped out, and kidney inflammation is pretty much cured. So that also has a lot of translational relevance, because several companies now develop antibodies depleting PDCs and so on. And then this interest in autoimmunity was further reinforced, again, through our basic studies of gene expression in dendritic cells. We became very interested in these particular DNAs. And for a while, we were studying it without sort of really knowing what to do with it, until really the human genetics solved it for us, because human patients were described with DNase 1L3 mutations that essentially developed very severe childhood autoimmunity, lupus-like disease. And of course, then with all our mouse work, we were able to confirm that the same is true in mice, that the deficient mice also develop autoreactivity, immune inflammation, kidney damage, and we were able to use this as a model to study fundamental mechanisms of autoimmunity and the substrates. So I'm stopping here. This is my lab still running, and hopefully it might give some sort of message of hope that you can make a lot of mistakes and still somehow things would work out. So just to briefly summarize the lessons learned so far, if anything I learned from my mentors is that to be complete, you have to be a good scientist and a good human being and a generous and supportive mentor. If Phil Leader could do it, anyone can do it. Mentoring younger generation graduate students and postdocs is the single best experience and also the single best investment into your science, because they are the ones who really make it work. Collaboration is good and sharing is good. And somehow things may work out despite all the circumstances and despite all the mistakes you can potentially make. It's better if you don't, but anyway. And as long as you just go and do what you really like, it's fine. Again, I don't think I'm fully qualified to make these strong statements, so I'd like to sort of bring in the higher authority. This is John Kendrew, who described the first crystal structure of a protein, myoglobin. So when I was a graduate student at the Weizmann Institute, he came to visit as an advisor, and our teacher brought him to our protein structure class. He was quite old and frail at the time, but he gave a very short talk to us. He said, you know, when Max Perutz and I were trying to determine the structure of myoglobin, everybody was saying, like, forget it, it's not going to work because this molecule is too complex. You cannot solve it by crystallography. But even if you do, why would it matter? What would be any sort of application of this in the real world? So, said he, don't pay any attention to what people tell you, and don't worry about translational applications, because you just do your science and all of this will come. So with that, I'll probably finish, and thank you for your attention.